Well, first I'd like to thank the organizers, in particular Ashita, very much for reaching out to me and for suggesting this and putting all of this together. It's really an honor to be invited, and I'm so glad all of you were able to make it today, despite daylight savings time. So today I'll just give you a little brief background about myself. My name is Julita Ramirez. I am a principal scientist at Pfizer at the Pearl River site and the Department of Comparative Medicine, and that's a part of global science and technology. And so a little bit about me, my background and history before we get into what I actually do. So I was born in Seattle, Washington, and I grew up in Lafayette, Colorado, which is a small town that's about 20 minutes outside of Boulder. So you can see the mountains, as you can see in that picture from there, but it is on the plains. So my education, I will start with college. 15 years starting from college. So I got my bachelor's of science in biomedical engineering in 2001 at Yale University in New Haven, Connecticut. And my specialty within biomedical engineering at the time, they had 3 tracks. That was electrical engineering, basically electrical, chemical, and mechanical engineering. As they apply to biomedical engineering. And so because at that time the program was brand new, and so basically you decided which way you wanted to track, and then you took mostly classes in that department. So I was chemi track, so I took mostly chemi courses. So that's Yale. And so that took 4 years. And then I went to Colorado State University from 2001 to 2008, where I got my DVM. That stands for Doctor of Veterinary Medicine. So I'm a veterinarian. And my PhD, my doctorate in biochemistry and molecular biology. And the story of how I actually got into that was very interesting. So in college, I went into college not knowing at all what I wanted to do. Initially, I always knew that I was interested in science, particularly the life sciences. Ever since I was a very little girl, I had just been drawn to that section of the library. I enjoyed watching nature shows and documentaries. I just loved science. And so then in high school, I attended a lot of engineering camps, and I thought I wanted to be an engineer. Because I didn't have a whole lot of exposure to people who went to college. And I thought, oh, well, if you go to college, you want to choose something that you need college for, and you want to choose something at the time, I really didn't care for school. I pretty much hated it growing up, mostly because I was bored all the time, and it just seemed like I could learn more on my own because I read all the time. And so I felt like I was wasting my time at school. So I only wanted to go to college for 4 years, and I was like, then I'm done, and I'm gonna get a job and start earning money. Well, so that's actually why I chose engineering, because I hated math. I was good at it, I just didn't care for it that much, unless it was applied. I loved applied math, but I didn't really care for the theory at all. So I went into engineering thinking that, like, oh, well, engineering is something that you can have a great career at with just a 4-year degree. And then I found out during college that I really didn't care for engineering. I did fine in my courses, but I always spent my time on other things. And my roommate noticed that, and she said, well, why are you spending so much time on your other courses and not on the courses related to your major? And she said, you don't really care for that. Why are you doing that? And I said, well, because I need to have a job after I graduate. And she's like, no, you need to do what you love. That's the most important thing. You need to do what you love. And that concept was foreign to me, because no one I knew ever, where I was raised, you got a job and you worked. And that's what you did your whole entire life. And then that was it. And I never sort of connected the concepts of, oh, you can, like, your work doesn't have to be just work. It can be a career. It can be something that you enjoy doing. It can be something that you love. And there were quite a few different things that I loved. So I actually ended up tacking on a second major, and it doesn't show it on this slide, a second major of molecular biology, actually, And so because I was more interested in the biochemistry and molecular biology, I was really into that. And so I tacked on that second major. And then I was actually, and at the time I still didn't know what I wanted to do, but I knew, okay, I don't want to be an engineer, but I know I still love science. I know that that's something I want to do, but I don't know what. So I was like, okay, well, I love animals. I've always loved animals. Maybe I could be a veterinarian, or maybe I could be a chemist, because I did love the chemical part of the engineering. I just didn't love the engineering part. So I love the chemistry. I just didn't love the engineering. So I was like, okay, well, maybe I'll be a chemist, or maybe I'll be a veterinarian. I don't really know. So I applied to graduate school for chemistry, a PhD in chemistry, as well as veterinary school. And it turns out that I got into a PhD program in chemistry at University of Colorado Boulder, but I didn't end up going there because I found out after the fact. I even accepted it and everything, and I was going to go there. Then I found out after the fact that I got accepted into the veterinary medicine program at Colorado State. And then at the time I was like, well, why do I have to choose? Maybe I can do both of them. And so once I got into the veterinary program, I looked into whether they had a biochemistry and molecular biology PhD program. They did, so I applied to it. And I separately was enrolled in both programs. So I did a dual degree, but it was a dual degree of my own making, basically, that both the departments just knew that I was doing that, and I sort of designed it myself, going back and forth. So it took me from 2001 to 2008 to do that, which is actually not pretty fast, considering that MD PhD programs, which I sort of designed my DVM PhD program off of the MD PhD, but basically I did first year PhD, then I did first 2 years of vet school, then I took 2 years off to do full-time PhD, under the assumption that I would finish my PhD. But of course, your PhD takes however long it is going to take, and you cannot say, oh, I will finish in X number of years. No, you finish when you finish, when you get a paper. But anyway, so I didn't finish in the 2 years, but then I was supposed to go back to vet school for my last 2 years of vet school, because vet school is 4 years, just like medical school. And so I ended up doing that, and finishing my DVM, or my PhD, it was like senior year of vet school. So I finished my PhD in 2007 and vet school in 2008. And that's Colorado State. Oh, and my project for the PhD was transcription of human T-cell leukemia virus, with Dr. Jennifer Nyborg was my mentor. And I really love biochemistry and molecular biology. So it's really good that I ended up doing that. And then my career took another fork, and I was like, okay, well, what do I do now? I have these 2 degrees that are kind of totally different. So what do I do? And again, I applied to a lot of different things. I applied to internships in veterinary medicine. I applied to postdocs as well, because postdocs are typically what you do after your PhD. And so I got into a postdoc program at National Jewish Health in Denver. And that's a respiratory hospital, a human respiratory hospital. They're famous for their work in TB, and they have a really great immunology department. So my postdoctoral training was in immunology, and my project there was transcriptional repression of B-cell genes, cell line and mouse models. So basically, I still did a lot of biochemistry and molecular biology, but as it is applied to immunology. And that was great, because immunology was another thing that I learned about in vet school that I really, really loved. In part because I think the reason I like molecular biology and immunology so much is because they require a lot of imagination. And I love to imagine things. I love reading. I love art. I love creating things, and that really is a lot of what you do, and you have to imagine things because they're so little, right? And immunology in particular is so complex, so it really does require a lot of imagination. So that's one of the reasons I love them so much. So, and it's also very difficult. So you feel like you feel like you're challenged, and so that's another thing I really like, to be challenged. All right, so from there, I went on for another 5 years to do a lab animal medicine residency at the Department of Comparative Medicine at the University of Washington. So I went back to Washington. They have 35 primary and affiliate veterinarians and 8 veterinary technicians. and 7 central vivariums with mice, rats, rabbits, pigs, dogs, ferrets, fish, frogs, birds. Yes, bats. We did work with bats as well. Big brown bats, actually. Guinea pigs, hamsters, and non-human primates also. So lots of different animals. And so my research projects would both use mice. I'm really a mouse specialist. I work with mice mostly. But it was called basically the functions of FNP1 protein in B cell development in tumors. So most of my experience and background in research in immunology is with B cells. Those are the cells that make antibodies. And the spontaneous loss of B cell transcription factors, there were 2 projects that I worked on, leads to pre-B cell leukemia. So that actually was a project that I had received a KO1 Research Career Development Award for the National Institutes of Health. So that basically funded, so it's a 5-year award that funds your salary for those years. And it's great because in research, there's not a lot of money at all. And so, especially in academia, so if you don't have funding, it's no matter how good you are, it's very difficult to find positions a lot because there's just not funding. Hopefully now with COVID, there'll be more focus on science, but in those days, there were lots of investigators in basic science that were losing their jobs. People who had been successful for years and career, just brilliant scientists, and they were like opening restaurants and things because there just wasn't the funding. And people, so basically, if you didn't have a grant to fund your salary, you couldn't continue in the sciences. So you had to pay yourself and your own benefits and everything else. And that's how it is. I don't know about now, but that's how it's been up until COVID. It's a wonderful, I love the life sciences, but it is very difficult to get funding, even if you're super smart and really great. So next, Taconic. So then from there, I was looking for jobs and I was looking for anything I could get because my funding had run out. And when your funding runs out, you are unemployed because no matter, again, like I said, no matter how great you are, oftentimes your mentors don't have the money to support you. They barely have enough money for your own research. Heck, I was from my own salary buying reagents. That's how it was. So, so I needed a job to make money to support myself after that funding wore out. So, pay rent and all that. So I was a veterinarian for the Germantown site of Taconic Biosciences. That's in New York. And they're a mouse and rat laboratory animal providers. So they breed mice and rats, genetically engineered mice and rats, as well as just the standard laboratory ones, only for medical research, not for any other purpose. So what I did there is I provided primary veterinary care and health testing. They had 35 different active animal facilities. As I said, genetically modified, notobiotic, that means noto is known, biotic, biome. So that means animals that are germ-free or they have defined flora, meaning they are infected with something, but we know exactly all the organisms that animal has. So known biome. And then also severely immunocompromised rats and mice. So I also had regulatory responsibilities. So medical research is very highly regulated, as it should be when it comes to the use of animals. You can't just do whatever you want, which is good. So to ensure, of course, that the studies are ethical and that only studies that really do need to be performed in animals are performed in animals, and also that the least sentient species is used. So for example, if you can do your experiment in yeast or fruit flies, that's what you do, and you don't use vertebrate animals unless your experiment requires that in order to answer your question. So, so for regulatory, I developed humane endpoints for disease models, adverse phenotypes, designs and implement studies for characterization of unexpected phenotypes. So for example, if you have a mouse that is genetically modified to have a mutation that, for example, makes it susceptible to breast cancer, of course, in mice, it would be mammary cancer. That's the correlation. But then you have to monitor those animals. So say, for example, you have a mouse with a BRCA1 mutation, just like a person, you need to monitor that mouse so you know when it's early stage developing cancer, so that way it doesn't get sick. And anyway, so that's kind of what that has to do. That's what an adverse phenotype is. And then for training, I trained operations staff in animal welfare, sick animal recognition, and technical procedures, and also directed the technician certification program. So I also had roles with customer service. I directed animal condition, customer complaints, customer consultations for creative solutions if they needed that, and colony management, and then audit. Customers would come to our facilities and basically look at everything to make sure, that everything was going according to their expectations, basically. And we also were certified by international accreditation, as well as various domestic organizations that regulate the use of animals in research. So then now I have worked for Pfizer only for 1.5 years so far. But I work, as I mentioned, at Pearl River, which is our vaccines research site. I do collaborate with colleagues in the vaccines research unit to design and refine models for preclinical testing of adjuvants. Adjuvants are the things that you add to vaccines to elicit immune response or to increase the immune response. Usually, so one thing, alum, for example, is an adjuvant, or it's usually some kind of chemical irritant that's non-toxic, but that does irritate to cause inflammation, because you need that component, since whatever it is that you're vaccinating with isn't virulent, meaning it doesn't actually infect you. You need something to turn the immune system on that says, hey, recognize this, and that's what an adjuvant does. And so then the immune response also, and to evaluate vaccine safety and efficacy as well. So that's what they do. What I do, I evaluate, recommend, and facilitate use of non-invasive in vivo imaging modalities. So for example, in vivo imaging systems where we look at fluorescence. So for example, if we have if we're working on an E. coli vaccine for urinary tract infections, then we could look at bacteria that express firefly luciferase, so that basically it's bioluminescent, and we can infect the animals with that, so we can monitor how the infection proceeds and whether or not our vaccine actually works to protect them against getting infected, that kind of thing. Because it's very important that our animals not get too sick. So that is sort of the that's why I'm doing that. And also refining techniques and reducing animal uses. So basically, if you are getting more data that is more reliable, and you can really follow the animal's health better, then not only are you taking better care of the animal and keeping them healthier, you also, because you're getting more data and more reliable data, and it's more accurate, et cetera, that you also can reduce your animal use that way. So it's kind of a win-win. And also to perform studies to validate animal models, smart cage animal monitoring, so that you can actually look at the respiratory rate, the temperature, the motion, the activity of animals like the mice as they're running around their cage and using their little wheel and stuff. So there's automatic monitoring, so you can know if anything's wrong. And also imaging modalities, like I mentioned before, because we do all the imaging modalities that they would do for a person who is sick. We also can do that for the animals who are sick. Even actually, our animals, I always joke that I would rather be in our animal facility than in like a lot of communities, because our animals get better care than a lot of people do. And they have modalities available to them like PET, CT, imaging, things that even people don't often get when they should. So, so that's really great. And then of course I train staff also. So for my last slide, I kind of wanted to give an overview of common types of vaccine studies in animals, just to sort of give you a 10,000 foot high level, high altitude view of how animals are used in vaccine research. Well, to begin with, most animals used in vaccine research and in research in general, biomedical research in general, most mammals that are used in biomedical research are mice. Zebrafish are used also a lot. And of course, that's not counting yeast and Drosophila, which are probably used much more. But yes, definitely mice are without a question used by far the most. They're like 98% of all research animals used across the globe are mice, at least vertebrate, mammalian vertebrate research animals. So there's the immunogenicity study where we vaccinate healthy, naive animals, and then we don't ever make them sick. We just vaccinate them, and then we study how their how their system responds to that, or the vaccine, the whole vaccine, or the individual vaccine components. So for example, if you're testing different adjuvants to see which one works the best. And so then basically we would, for example, bleed the animals to see what was going on there. For safety study, we would evaluate any harmful effects observed. So as a result of the vaccine or drug administration, those are usually performed in rats. And they're not usually actually done at our site, but that is something that is done for every product because it's required to be done in order for those products to be used in people, to be licensed to use in people. They can't ever be licensed by the FDA or by other global regulatory organizations unless those studies are done and have been shown safe. So then the challenge studies where you vaccinate healthy naive animals and you inoculate them with a disease agent. So that's where you actually challenge them. You don't just vaccinate and look at the response. You vaccinate, then you challenge them with whatever the agent is and see if the vaccine can prevent them either from infection, that's the level of protection, from disease, otherwise known as morbidity, or from death, mortality. So that's what I mean, different levels of protection, right? So you could, like, the best is where they don't get infected at all. The second best is where they might get a little sick, but not really that sick. And of course, the worst, it's still protection, but they get really sick, but they don't die. But we would rather that they have better protection than that. So, and animal model selection is animal model, basically the species that we're using is based on the similarity of the disease and or the immune response, and also the lowest phylogenetically advanced animal that we can use that still gives us the answer that we need, that we're looking for, that still is a good model system. And that's one reason why mice are great, is because they're small, and their immune systems are actually very much like people, more so than even more advanced mammals. So that's one reason they're used a lot as far as mammals are concerned. They're used the most. So I will actually that's the end of my part, and then I will take the questions.
All right, thank you so much. So we'll hop into the questions. Okay, so how did you decide to pursue a career in your specific field?
Yeah, so that one, I sort of alluded a little bit to it, but basically because I had really no idea of what I wanted to do. I knew there were a handful of things that I enjoyed. And so I just kept trying things until I found things that I really enjoyed. And then, I love the biochemistry and molecular biology, and I was a veterinarian, and I said, well, what can I do with these 2 degrees? And really you could be a lab animal veterinarian or a pathologist where you look at slides and read out slides all day long. And I didn't want to do that because I'm a more active type of person. That's actually one of the reasons that I like molecular biology so much, is because it's a lot like cooking. Like, you do your dishes, you do your assay, you have your protocol, you do everything, and you're running around all the time. You're constantly active. I mean, I would do like 15, 20,000 steps a day when I was in lab because I was just running constantly the whole time. And so being a pathologist, even though I really like pathology and I think it's very interesting, it's too much sitting for me. I couldn't take it. So I decided, okay, I'll be a lab animal veterinarian. And I did, in veterinary school, I was more interested in internal medicine really than I was in surgery. And so I decided basically to be a lab animal veterinarian because combined, it was the field that best combined my expertise in science with my expertise in veterinary medicine, and taking care of animals, and also getting to work with such a wide variety of species. Is I think the biggest two advantages of being a lab animal vet, the biggest advantage actually for me, is that our animals get better care than a lot of pets do, because people can barely afford, a lot of people can barely afford to take care of themselves, unfortunately. So, you know, how are they going to take better care of their pets than they take care of themselves? I mean, they might, but my point is that we spare no expense for our animals. Money is never a reason that animals don't get treated. It's actually against the law. So, you know, when you're a veterinarian, there's a lot of us that get burnout because we see a lot of animals that are sick, and we would like to treat them, but we can't, because we can't give free care to everybody, and it costs just as much, you know, for the medical equipment and procedures on an animal as it would for a person. And so that's one reason that suicide is so high among veterinarians, which is actually a big problem, because we're really empathetic. We care so much about animals, and it's really hard to see them die when we know it's something that we could treat. And that doesn't happen in lab animal medicine. It 100% does not happen. So do animals die? Obviously, yes, but they're euthanized in a humane manner if they're sick and they can't be treated. And if they can be treated, they are. So, and we as veterinarians get the final say. If we say this animal needs to be euthanized or this animal needs to be treated, it is. And so that's the biggest thing for me, is that really I feel like my patients get the best. And the other thing is that I get to work with a bunch of different species and solve challenging problems. So yeah, it's nice that way. I mean, obviously there are tough things too, right? Because there are some branches of lab animal medicine as really hard, like for example, toxicology, where they are giving you know, different doses. They basically ramp up to see where the toxic dose is. Those studies are really hard. And so thankfully I don't work with any of those. But pros and cons.
Thank you. Okay, so our second question is, how has doing research changed with COVID-19?
So doing research with COVID-19, I think research, so basically most researchers, if you're doing assays or you're actually doing an experiment, you still have to go into the lab. So research personnel are semi-essential because they do have to go in, but it depends. It can be hybrid too, because if you're analyzing data or if you're writing a paper, for example, you can do that, or giving a presentation like I am now, you can do that obviously from home. So a lot of lab workers have hybrid workers. So basically for all their experiments, they're in the lab, in the office. But if they can do it at home, then they're at home. So it's hybrid. So that's how it's kind of changed physically, the physical aspect of working. Although I will say that research has always been a field that's been more flexible than other careers. So depending on whoever your lab head is, of course, but it's kind of been that way a lot even before COVID-19. Of course, COVID-19 made it very much more so that way. The other thing that I think COVID-19 has two other things it's really done is, one, increase public awareness and appreciation of research that we didn't have before. You know, there really was a lot of opposition to research or people not understanding it. I mean, I think that still exists, but I think now people see, instead of seeing, for example, vaccines as something that gives people autism, or, you know, which is not true, by the way, but instead of these sort of scare stories or researchers, these white coat people who like torture, who do like horrible things, they're realizing, hey, no, actually, science is towards the betterment of humanity, right? So we're discovering things, we're being curious, we're asking questions, we strive to be highly ethical for the highest standards of mom. I live with my mom. Efficacy. So yeah, so it is. It's nice that people do understand a little bit more, and give us a little bit more recognition. And also collaboration has really increased. Again, I think science has always been an area where collaboration has been pretty good for the most part, and more so than other fields. Even if you're in industry, there's a lot of collaboration within the organizations and even outside. But with COVID-19, even more so. So that's great. So the people are really sharing information more and talking to each other more. And those are two really positive aspects that I see is coming out of all of this.
Thank you for pointing that out. Okay, we'll move on to question number 3. Do you have any advice for high schoolers or college students who want to pursue research?
Yeah, I would say you be um, just be patient and try a lot of different things. So tenacity is one of the greatest things qualities you can have as a researcher that you will need. So 90% of the things you do aren't going to work out, but the small, like 5 to 1% that do are going to be great. So just have patience, be tenacious. You know, if at first you don't succeed, try again about 1 million times. Setbacks. Yes, I would say for me, the biggest setback was not understanding about higher education, or not really having any role models or that kind of thing. Because I didn't know how it worked, how any of it worked, and what to expect. And so for me, that was the hardest thing, is sort of going into the whole process of even getting a PhD. It was a total black box. I really didn't know what it was. I didn't know how one did this or any of that. So yeah, so that was really the hardest thing for me to overcome, which is why I enjoy reaching out and talking to students, because I really feel like if I had known, it just would have helped me so much more to have an idea of what I was getting into back then, because I really had really no idea of how science worked, because I just didn't know anybody who was a scientist or had even a college education. So yeah.
Thank you for that insight. So how involved are you with the COVID-19 vaccine research, and are you familiar with how it's developed?
Yes. So our site is vaccines research. But we didn't do a lot of the animal studies because those had to be outsourced, because we don't have COVID-19, because it's such a severe respiratory disease and so transmissible, has to be worked at with biosafety level 3, which I won't get into the details, but it's highly regulated. And so in order to work with that, you have to have a facility that is that biosafety level. And so we outsourced all of our studies. But I will say Pfizer and BioNTech is the partner that developed the COVID-19 vaccine with Pfizer. We actually had been working for a couple of years already on an influenza vaccine with the same platform. And so we'd been doing a ton of research before then. So that's why we were able to switch over so quickly. All we did, because it's an RNA vaccine, so and manufacturing nucleic acids, synthesizing them is super easy and very highly scalable. So once the sequence was known, that's all we had to do. It was plug and play, literally plug and play. We just had to decide which of those sequences for the spike protein we were going to use, and test which ones were going to be the best. And that's why we were able to so quickly scale up, and we had already had safety studies for the vehicle and everything in animals because we had been working on it for influenza. So I think, and Moderna is kind of a similar story. So I think this technology, the mRNA vaccine technology, it was just lucky that COVID-19 came along, I guess, if it had to come along when it did, because that technology was ready. It was on the brink of being developed, of being prime time. And so it just launched it because people had been looking into RNA vaccines for, gosh, since I was in grad school, 20 years. So yeah, so it's exciting. And that's another good thing that the pandemic did amongst all the horrible things that it did, right? One of the good things was that it actually pushed the mRNA vaccines, which may have taken otherwise years and years more to become prime time and be established. And now we're so much better able with these now being tested right in the population. We're so much more able to, because the problem with vaccine research is that it takes a very, and the reason why you've heard on the media is it takes so long to develop vaccines historically, is because there is very little risk tolerance, which for good reason, very little risk tolerance with vaccines, because of course you're vaccinating healthy people. So therefore there pretty much can't be any harmful effects from what you're giving, because these people are healthy. It's not like a cancer patient that's going to die if they don't have some kind of treatment. So if the treatment makes them sick, as long as it makes them better in the end, right, that's a win. Whereas with a vaccine, we can't have that. So so previously the risk aversion to vaccines was such that it just took a really, really long time to develop. And people, regulatory agencies, nobody wanted to fund studies, regulatory agencies didn't want to approve them, that sort of thing. So, oh, let's see. Do I see a risk of autoimmune disease as a possible long-term effect? No, 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 I don't. Because of the fact that so it depends on the vaccine you're using. But so mRNA vaccines use lipids. It's basically a lipid nanoparticle, like a micelle, and then it has RNA inside that codes for the protein, so your cells make the protein, and it's a spike protein. It's, in the case of COVID-19, it's the viral spike protein. So that protein is very unlike any that we see. I don't see a big risk for autoimmune with the RNA vaccines. It is known that other types of vaccines, and depending on what the antigen is and the platform, have been known to cause autoimmune disease. So it's a very good question. Of course, in a very small percentage of the population, but yes, for example, because influenza is linked to some of these syndromes, like the Guillain-Barré syndrome. So, and how do I advocate for animals' welfare and research? Well, it's my job. That's 100% what I do, and the other veterinarian. So I'm not even the clinical veterinarian. Because at my site, even we have two clinical veterinarians and also another veterinarian who basically runs and manages the facility. But basically, it is our job to, as well as our regulatory colleagues, because we have people whose job it is to review the animal protocols and to review all of the research that's proposed. And right, you can't ever do any research until a committee meets, including outside members. There's a whole process, and which is good, right? That's what it was designed for, is to protect animals. So yeah, so I advocate for them by making sure that we use as few animals as possible, making sure that the studies are appropriate, that questions can't be answered any other way, that the animals have care, that I get the most advanced imaging modalities, which we actually, so we are getting, for example, we're getting micro CT for rodents, rodents to ferrets. And that's because we're actually going to be opening a new BSL-3 suite at my facility so that we can perform research on, for example, pandemic strains of either the flu or COVID moving forward. So we can actually perform it there. And so ordering a micro CT that's the best and the most advanced for all of these species. So it's basically like staying at the cutting edge of medical technology, human medical technology, so that way our animals can get the very best care. So I hope that answered.
Yeah, thank you. So now we will be having our audience Q&A. So feel free to ask questions in the chat. Christina will introduce the questions.
Okay, so we have a few questions in the chat that have already been answered. So I'll be trying to pick out the ones that were not covered as much in the Q&A section for the slides. So the first question was, why did you decide to start working for Pfizer, and what does naive animal mean?
Oh, yes. So Pfizer, I decided to work there because actually my friend, Artie, who's also a veterinarian, we went to the same residency program. He was working for Pfizer for a couple of years and he really enjoyed his job. And so when they had an opening, he actually called me up and said, hey, you know, are you looking for someplace? Would you like, you know, to apply? Is this something that would be interesting to you? And I said, yes, you know, this position looks very interesting. So that's why I decided to apply to Pfizer. And also I had kind of wanted to work in industry because for a long time, because I felt like in academia, it's a great place to learn and you have a lot of questions that you can answer. But the funding is so bad and so hard. And I had seen so many stories of people who were so highly talented and just couldn't make it. And whereas you're less worried. So when you work for a company, they pay your salary and it's your job to do your job, not to pay yourself. So if that makes sense. So because in academia, you would spend almost all of your time writing grants and much less time actually doing science because you were so focused on getting money. And so it seems odd, right? Because people think of corporations as like, oh, they're money hungry, they're powerful. But on the other hand, if they have money, they can pay a decent salary to their people who are talented so their people can really focus on what matters and can bring these things to the public. That's the other thing, is because in academia they have these awesome ideas and people who are so talented, but a lot of times the ideas die because nobody else gets on board with them and there's not the money and they don't have like the resources and they don't have what it takes to get them, you know, to get their wonderful ideas out to the world. And that's what you have in a corporation. They have all that money and power that actually can get things done. So that's kind of why I wanted to end up working for a company, because I wanted to see change happen and I wanted to improve people's lives in a very substantial, like in a way that I could actually see and feel like I was contributing there. So, and then I think there was a second part of the question that I did not address.
Yeah, the second part was, what does naive animal mean?
Oh, naive, yeah. Just an animal that has not, naive has not seen whatever the antigen or the vaccine is. Or so, for example, I am naive from COVID if I have never had COVID and if I've not been vaccinated. But once I've had COVID, I'm not naive anymore, if that makes sense. Naive to the agent and naive to the vaccine.
Okay, awesome. Thank you for answering those questions. I was wondering what naive meant myself.
Yeah, sorry, that's it's a immunology term that you get so used to how we talk. But yeah, it's they don't know yet. Your immune system is naive. Their immune system has not met the disease agent or the vaccine.
Mm-hmm.
And stranger.
The second question, I think you already answered a little bit of it as well, was what would you recommend I do now if I want to go into biomedical engineering or immunoengineering? can I/should I double major in those fields?
You can. I mean, I would say to people, sort of just follow your interests. And while it's, I guess, maximize your time and do as much as you think that you can. Squeeze in there while you're in high school and college without, going crazy or being unhealthy or things like that. You still need to work hard, play hard, right? You still need to have fun and have friends and all of that. That's super important. But I would say yes, double major in whatever your interests are. Think about creative ways that you can combine them to fill a niche that maybe nobody else has. So for example, if you love photography and you're really interested in immunology, maybe, or some other kind of science, maybe you could become or art, you could become a biomedical illustrator, or you could work for a journal, or you could, be an editor, or, there's so many careers, or you could create your own career too, based on like the intersection of whatever those two fields are, or three fields, right? Even so whatever it is that interests you, just try to find those aspects and keep your mind open and be creative. That's something that really helped me too, is the more people you know and you meet, be like, oh, so-and-so did something like that. And like, oh, well, what if I did something like so-and-so, and then this other person did this, and what if I could do that too? So yeah, I would say kind of maximize your potential, and if you're interested in it, do it, explore it and see where it leads you. And I think that's one of the great things in life, that you can really be curious and explore and go where you want. My experience for my DVM, well, it was fun, but it was also, well, it's a lot like medical school. So it's 4 years. Your first 2 years are classes, and your last 2 years are practical. And so, yeah, it's a lot of fun. It's a lot of work. But I would say a PhD is harder. And it's harder to get into vet school, but once you're in, you just, you do the work. You just do, if you're good at academics, you just do the work. Whereas with a PhDs are very difficult, because you're not guaranteed to get one, no matter how smart you are, no matter how gifted you are. You have to be lucky also, and you have to be a hard worker, all three. Because and so I'll tell you, and it's really, really important, your choice of mentor, really important. You have to see how many people that person has graduated. So that's another thing I would say when you're choosing your mentor, if you're going for a PhD, make sure you choose it. Don't choose based on the project. I've had so many friends that fell down this hole. It's not good. They would get fascinated by a project that be like, oh, this project is so cool, I want to work on it, no matter who the PI was. And they'd be like, oh, the project's really cool, I'll be fine, just because I really want to do the project, which seems good, right? But it's more important that you work on so for your career, you want it to be something you're interested in, right? And interesting. But for your PhD, you just want to be able to get published so you can graduate, because graduating is it's not like anything else in your life that you've experienced. I will say you can't just, take some classes and do the assignments and get good grades and pass. That's not how it works. You basically have to do a ton of experiments in the lab. They have to lead somewhere, and they have to be and the answer has to be interesting in a way and you have to not get scooped by other people, meaning nobody else can publish what you are working on before you, or else guess what? All your work for nothing. Because most programs require that you have at least one first author paper in order to graduate. So you want to choose a mentor who has a history of picking projects that can get people graduated and that really helps their students. And of course, who is ethical, and because that's another thing you might not think of, but it's really important that you agree with your mentor on their ethics. That you think they are good and that they are a good person and stuff. That's really important. But yes, if you do that, even if your project is boring, or if you think it's like more boring, that's okay, because at that phase, what you're focused on is learning techniques and getting published and graduating. So learning the concepts, learning the techniques, getting published and graduating. Then you can go on afterwards and work on the really super cool stuff. And because you're not worried about graduating anymore, then it's fine. But yeah, before then, just graduating is more important than projects that don't pan out or that get scooped. I've had too many friends who actually left science because they spent 5 years trying to even get a master's and then didn't. So just keep it in mind. It's not guaranteed, even if you are smart and a hard worker. So.
Okay. So our next question would be about Pfizer again. What do you do in a typical day at Pfizer?
I guess I wouldn't say there's really a typical day, which is one of the reasons I chose this career. But I do have a lot of meetings. So I guess I would say meetings are, and most of them were virtual even before COVID, because I collaborate with a lot of people, mostly in the US, but across the world too. So I have a lot of meetings about all kinds of different various things. Some of them are listen only, a lot of other ones are participatory. And then the other thing is I'll go in for procedures. So for example, if the clinical veterinarians are, have an animal that has some issue, I'll go in to help image. Or I might go in to teach people how to identify lymph nodes, or I might go in to harvest muscle tissue or teach a technique to somebody or evaluate how people are spinning down their samples or all kinds of different things. And so that's another reason why I really enjoy this kind of career, is because it does have so many different aspects to it. So you're not just doing the same thing all the time.
Awesome. The next question is about the vaccines. So they're wondering if what you're saying about when a person gets sick from a vaccine, does that mean the vaccine does not offer as much protection?
I guess it depends on what you mean by get sick from a vaccine. Do you mean by side effects? Like if you have side effects from a vaccine, or so there are different kinds of side effects. So for example, with the autoimmune question, there are certain antigens that and certain reactions that people can have to injected substances that if those substances look enough like some parts of our body, they can develop autoantibodies. That is, antibodies that also bind to parts of their own body that are not good and that elicit an immune response, a self-directed immune response. So that's kind of separate, because autoimmunity is it's a chronic condition that is not easily treatable. It is treatable in many cases, but not most of the time it's not curable, although sometimes it does resolve spontaneously. Versus, for example, the kind of side effects that you see from a vaccine, like, for example, like temporary side effects, for example. So we're not talking, so long-term versus short-term. So temporary side effects like, oh, my muscle is sore, or, I have a fever, I got a fever or something like that. Those temporary side effects, actually, the more that you get, the better potentially, because that means that your immune system is really recognizing and being activated by the vaccine, which it is designed to do. Current vaccines for COVID that are on the market, at least in the US, the three that are approved for emergency use, which are the Pfizer vaccine, Pfizer-BioNTech vaccine, the Moderna vaccine, and Johnson & Johnson. All three of those vaccines, none of them actually contains COVID-19 virus in any way, shape, or form. They contain nucleic acid, either in the case of Johnson & Johnson, so they have the lipid particles, right? Those little fat droplets. And then they have inside, they either have mRNA, so Moderna, Pfizer, the ribonucleic acid, or they have DNA, in the case of Johnson & Johnson, that encodes the spike protein. So that nucleic acid, whether it's the DNA or the RNA, gets into your cells, and your cells make the spike protein. So there is no virus involved. And that's another reason why they can manufacture so many doses so quick, is because, so for example, influenza shots, you have to actually grow the virus in eggs for a long time, and then you purify it, and then you inactivate it. And that's what you inject people with. So that's a different kind of virus though. So, but these vaccines for COVID that are currently emergency use authorizations, they don't grow the virus. They don't have anything to do with the virus. It's all nucleic acid based. So that's why it can happen so fast, and why you can have that, you know, basically all you need is the sequence for whatever protein it is you want to make that you want the immune system to recognize. Does that make sense?
Yeah, I hope that answered your question, your most recent specification, Mary, about that, because Mary was specifying that after the second COVID vaccine, she felt very sick. So
That's
with,
common.
yeah, okay.
That's common. And and it's common, I'll say it's common in young people, because young people have a more vigorous immune system. So for older folks, it's not as common because they don't have naturally your immune system efficacy declines as you age. So it's very normal for people to have, especially after the second, with this kind of vaccine, a very strong response, especially if they are healthy.
Mhm.
Yeah, and that's one of the reasons why the vaccines out there work so well.
Awesome. Another question is, how would you explain vaccines to younger children, like children over the age of 5? Are there any analogies you would use while talking to them about vaccines, or for people who are not familiar with scientific terms?
Yeah, I guess I would say that a vaccine teaches your body how to protect itself. It teaches your body who the bad guys are. Because before your body doesn't know which guys are bad and which guys are good. But the vaccine shows you the bad guys, but the bad guys are already dead, or maybe it's just a picture or a book. In the case of DNA, it's a book about the bad guy. It tells them, it teaches them, your body, it teaches your body how to fight, who the bad guy is, and they already know how to fight, but it teaches who they are so they can recognize the bad guys and fight them, the disease.
Mhm.
And that can keep you, if it's good enough response, it can keep your body from the bad guys ever even getting in.
Awesome. Yeah, I think that makes a lot of sense for kids who like don't really know what a pathogen is or something. They can just see them as like villains or yeah.
I've seen like some cartoon depictions of your immune system as like a shield, and then all these little bad bugs are like coming, and the immune system is like a sword and a shield, and is defending and is killing, and yeah.
Mhm. Awesome. So another question I think you already answered in the slides, but one from Ashley is, what was your experience like during the time you worked towards a doctorate of veterinary medicine and a PhD in molecular biology? Do you have any advice for students such as herself who aspire to become a veterinarian and perform animal research?
Yeah, so I would say to be a veterinarian, just make sure you have enough animal experience while you're in high school, so that get a job working with animals. So whether you're a trainer or behaviorist, maybe you work at your kennel, or volunteer, right? It can be like a volunteer position too, that's fine. Or even in research, if you can find something where you're volunteering in research, that's great, but it doesn't have to be in research. You know, you can volunteer at an animal shelter, you can, you know, at a handicap riding center with horses. There's all kinds of things you can do, but you need a certain, like for me, every school is different. So you should like look at the veterinary schools and what their entrance requirements are. And at Colorado State at that time, you needed 1,000 hours of animal experience in order to be considered. And then of course, yes, you have to take certain classes and get certain grades. And I feel though like most high schoolers are super familiar with and understand the concept of like, oh yeah, I have prerequisites and I have classes that I have to take and get a certain grade. Everyone, we get that. You guys get that. But that's the thing they don't think about is, oh, practical experience is really important too. So I would just say make sure you have that. And I mean, other than that, it's just what you do for success in your regular life. You know, you the same kind of formula for succeeding in high school too, is just working hard and time management. And time management is really a big deal. You want to make sure you can focus on the things that are most important and do those first, because the more advanced you get in your academic career, you're going to have too many commitments that you can't possibly do them all to the certain level that you want. But you have to be able to identify, okay, what are the absolute minimum that I can do, and then what are these and like the most important things to you. Like, what is the most important thing to me that I get out of this class? Because of course it's not just about passing, but if you're like, okay, this is the most important thing that I want to learn this skill, or I want to understand this concept, and just focus. Being able to focus is very, very important, because you're going to get overwhelmed. I mean, you're probably already overwhelmed because high school is crazy. But yeah, it's the same sort of thing to get through that. Yeah. And the experience, I guess, going through vet school, it's glorified high school. It's the same thing.
Another question is about the process of getting a PhD. So unless your research is published, is it true that you can't get a PhD?
It depends on your program, but in most programs that's true. And you can't get published. You can't just do research and get it published. That's another thing that people don't understand. It has to be something that is, basically you have to be telling a unique story that people are interested in hearing and that they haven't heard before and that nobody else has told. So I guess that's the best way to describe it. So yeah, so getting published is a very big deal, and it's not easy. It's very hard. And I would say that's the biggest thing to overcome. And some master's programs even have a publication requirement too. So it just depends on the program. So if you're interested in various programs, and they're going to be different between institutions also. So for example, the chemistry program at CU versus the chemistry program at University of Washington, perhaps totally different requirements. So just make sure that you understand the requirements of the particular program that you're applying to, because they are really different between programs.
Okay. So this is our last question. So the J&J vaccine is a DNA vaccine versus an mRNA vaccine. Is the risk of autoimmune disease better or worse than one or the other? And is the greater risk of autoimmune disease associated with the adjuvant rather than the nanoparticle?
Okay, well, honestly, I'm not sure if the J&J vaccine is adjuvated. That's a good question. So I don't know is the answer. From what I have heard, there, because J&J does use an adenovirus vector. It's an adenovirus that's harmless and that is present in the human population, that kind of thing. And so that's the vector, that's the actual package that gets the DNA into the cells. So the thing about the nucleic acid vaccines is that often they don't actually need an adjuvant, which is great. So that's one of the reasons why there's not as big of a risk of autoimmunity. However, autoimmunity is such a crazy beast. It's very difficult to predict. So I would say, so I can't really say, but in general, I can say that based on data we have from the clinical trials, it is, for example, in all three of those vaccines, it would be less risk, for example, than a flu vaccine, if that makes sense. So if your risk from a flu vaccine is like 0.01% of getting autoimmune disease, then it's less than that.
Okay.
Because to this day, I have no knowledge of even a single confirmed case of autoimmunity linked to any of these vaccines. Now, given, as we all know, they haven't been around that long, right? So something could happen. But so far, so good. Fingers crossed.
Right. Awesome. Then that concludes the questions that we have from the audience. Thank you so much, Julita, for being our speaker today. We learned a lot of great things about the vaccine process and like the PhD process and like everything to do with your major. And personally, I learned a lot. Like I didn't know that a biomedical illustrator existed. So
They do really cool stuff too. Yeah. So yeah, there's so many. I mean, that's the thing is, you know, there's so many careers. Just like make sure you sort of, you know, search for different things and talk to different people, because there are so many unique and creative careers out there. And if you can dream it, you can do it. And I know that's a cliche, but it's true.